Okay, so in the T cell dependent B cell activation, we'll divide it into the two parts here. One is the B cell and one other one is T helper. Particularly it starts with the TH0, that is the naive T helper cell. So we know that our antigen presenting cell present the part of antigen fragment on the surface here by the help of MHC class 2 molecule. And our TH0 means it's a naive T cells, T helper cells, when came into the contact with this by using TCR, right? CD28 and B7, no? So this we already know that this T cell activation requires two signal. Our first signal is all about, see, first signal is the binding of T naive cells with the antigen presenting cell. And our second signal, important signal, is binding of B7 with the CD28. So these two signals are very important for the activation and the differentiation of naive T helper cell. So after this naive T helper cell is now differentiated into the type TH2. Okay, so this TH2 is very important. So this TH2 is now activated. Meanwhile, this macrophage release certain kind of interleukins like 2, 1. This activated T helper cell. So this is your T cell. It is a activated C cells, and you will see the number of copies of this kind of activated T helper cell here. So this is the activate against this particular antigen. Suppose that is the antigen X. So activated against X epitope. Meanwhile, the same thing happens with B cell. So we know that B cell is having the BCR, and this BCR when came into the contact with this kind of epitope X, this is the first signal for B cell, and this is the first step in the B cell activation. But at this point, this B cell is not able to grow and differentiate into the plasma cells. Mind it well, this differentiation of this naive B cell into the plasma cell requires the help of other cells. And that other cell is nothing but the TH2 cells. No? So, what happened here? So, suppose you have a B cell here, and on BCR you have epitope X. At the same time, your activated T cells, no, it's a TH2 cells, it is your B cells, and this is the common epitope X. When these two cells came into the contact with each other, this TH2 cell now release various kind of cytokines like Ig4, 5, 10, 13, various kinds of interleukin cytokines produced by these TH2 cells. And this is the key chemical requirement for this B cell to differentiate into the memory cell and the plasma cells. Without the help of this TH2 cells, this B cell is not able to differentiate into the plasma cells. Once plasma cell, then that will produce a number of specific antibodies against antigen or epitope X. Is it clear? So that's why this whole pathway is known as T cell dependent B cell activation. So the bacteria, blood cells, when we encounter this kind of antigens in our body, our B cells came into the contact with this kind of antigen. Meanwhile, macrophage present the same antigen or same epitope on the MHC2 receptors. TH0 came into the contact with these two. This activate and differentiate TH0 into TH2. Here also the first activation, but this conversion, or we can say that the differentiation of these naive beta cells to plasma cell requires the help of Th2 cells. So binding of Th2 with the B cell leads to the secretion of variety of cytokines by the Th2 cells, and these cytokines will help B cells for the production of various antibodies against this specific X. Okay, so this is called the B cell activation, but that is the T cell dependent, right? So some interesting facts are there. Suppose this is the one naive B cells. It carries around about 50,000 copies of BCR on its surface and produced, once it gets activated, it will produce 10 million copy of antibodies. If you talk about the variety, 10 raised to 13 different types of antibodies produced by this B cell. Now imagine the importance of B cell here. Without the B cell, will not get any antibodies. So each cell is having, or each cell is carrying this much amount of BCR on its surface. So it is very easy for the cells to come into the contact with the different epitopes, right? Once it gets activated and converted into the plasma cell, round about 10 million copies of antibodies produced by this plasma cell, activated plasma cells, no? B cells. So this mechanism is very important. Apart from this, we already discussed another type of mechanism, that is the T cell independent. As I told you, it is not that much important for immunological response. Why? Because certain type of epitopes like LPS, I'll give you the one example, LPS, lipopolysaccharide. No? It's a polymeric antigen, so polymeric form. No, so these kind of certain polymeric molecules are able to activate B cells. So binding of these on the surface of B cell antibody here activates these B cells, produce number of B cells. No, this will activate number of B cells, right? But they are not able to produce antibodies, and that is why this independent cycle, independent B cell activation is less effective immunological point of view, because protection, if we talk about the protections against this kind of pathogen, we need our army of antibodies, and that mainly that was produced by T cell dependent B cell activation pathway. No, so this is all about the B cell biology. These are the very important points that I have covered over here. So once a simple recap, B cell mainly produced by the stem cell present in the bone marrow. This stem cell produce B cell precursor cell, B cell precursors to B cells. Then the maturation, and that B cells you will find it into your bloodstream, where these are the naive B cells because they never came into contact with any antigen till that moment. When this naive B cells encounters this part of antigen called epitope, it gets partially activated but not able to produce antibodies because it requires certain growth factors and the differentiation factors like interleukins. So that will be provided by T helper cells. So based on that, there are two pathways: T cell dependent and T cell independent. The most important is T cell dependent pathway. So for that reason, we need certain receptors. So B cell is having the B cell receptor on the surface of the plasma membrane, where that one B cell receptor is a complex molecule having one transmembrane antibody molecule, that is the M or D, and along with the IgM or IgD, it's having the Ig alpha and the Ig beta in the transmembrane part. All this complex is known as the BCR. So when your antigen means epitope binds here with this particular antigen binding pocket. This will activate B cells, right? And after that, this will activate endobiosis pathways. So the B cell also acts as a antigen presenting cells. So by doing this, B cell helps T cell also, and T cell helps B cell, right? So in this pathway, T dependent, we discuss the activation of Th cells, Th0 to Th2. This activated Th2 helps B cells by secreting various cytokines, and these cytokines help these B cells to convert it into the plasma cells, and these plasma cells will produce number of variety of antibodies against that specific epitope, and that will help us to provide humoral mediated immunity. And T cell dependent, mainly polymeric antigenic substances will activate the B cells, and that is the T cell independent. So no T cell is required here, but B cells gets activated, but no antibodies producing plasma cells produced or differentiate in the case of T cell independent B cell activation, and that's why it is less important for our immunological point of view. And certain facts like each B cell is having the 50,000 BCR, produce more than 10 million copies of antibodies, and variety of antibodies 10 to 13 different varieties of antibodies produced by B cell. So this is all about the B cell biology. Thank you so much. If you have any doubt, definitely you can contact me. You can comment it here. Your suggestions are welcomed, and I request all viewers, please subscribe this channel. Thank you so much.